Welcome to Frederick National Lab Technical Service video series. Today we have Dr. Steven Stern from Nanotechnology Characterization Lab. Dr. Stern, welcome and thank you for talking to us today about unique services your lab provides. Please tell us a little bit about services that you offer.
Well, thanks a lot for the opportunity to talk about the TSAs that we offer at the Nanotechnology Characterization Laboratory. Today I'm going to be talking about two TSAs, NCL01 and NCL02. These are both based on the Situa fractionation method. So the Situa fractionation method is the stable isotope tracer ultrafiltration assay. This is a fractionation method that was developed in collaboration with the FDA. To our knowledge, it is the only general purpose fractionation method that can be used in almost all different drug formulations to arrive at accurate estimations of encapsulated, unencapsulated, free, unbound, and protein-bound drug concentrations, all at the same time with two simple analytical runs on an LC-MS. Now, the method, as I said, is based on this Situa assay, which uses a tracer to determine the amount of protein binding in a system. And once that tracer determines that amount of protein binding, it's very simple mathematics to arrive at all the various drug fractions within that biological sample. So this can be used for aliquots from a drug release study in vitro, or similarly aliquots from a pharmacokinetic study in vivo. And in fact, NCL01 and NCL02 are based on those two different scenarios that I just described. So NCL01 is looking at in vitro drug release in human plasma over time. NCL02 is looking at a pharmacokinetic study in rats, looking at drug release over time in plasma drug concentrations for a nanomedicine or complex drug formulation. In both cases, we do a partial validation of the bioanalytical method used to measure the analytes, being the drug analyte, the API, and the stable isotope tracer. We then go on to run controls for the Situa assay itself to ensure the reproducibility, accuracy, and precision of the Situa assay, looking at spike controls, in-process controls, and various other controls to ensure the integrity and robustness of the assay. Following that bioanalytical validation and validation of controls, we then go on to conducting the study, which in the case of NCL01 is an in vitro drug release study looking at drug release at various time points. We can conduct that study within a 5-week time period from initial receipt of the formulation. NCL02, looking at pharmacokinetics in rats, again uses the same initial validation and control studies to then go on and perform the study in rats following validating the method. And that study can be conducted in an 8-week time frame upon receipt of the materials.
Thank you. And what makes these services so unique?
So what makes this service unique is that, to our knowledge, it is the only we are the only providers of this Situa method in the world at this time. There is no other commercial source. And for that reason, there's an extremely high amount of interest in our laboratory in performing this method for groups. There has been some talk recently of other providers taking on the method, but at this time, we're the only source of the assay.
Okay. And with that, what type of research or what type of researchers do you normally support?
So normally we support advanced preclinical and even clinical drug development. These are researchers often that are working on generic nanomedicines or complex drugs, or novel nanomedicine and complex drug formulations. So often this assay is utilized in clinical trials to evaluate bioequivalence, as well as preclinically to look at pharmacokinetic comparability to previous established drug formulations.
Thank you. Dr. Stern, thank you so much for talking to us today. And for those who are interested in learning more about these services, please do not hesitate to contact us. Thank you.
Thank you for giving me the opportunity to discuss these. Thanks.